Now we are going to discuss about syphilis, which is caused by a bacteria Treponema pallidum. Okay, this belongs to phylum Spirochetes. See, this Troponema pallidum is spiral shaped. Okay, okay. Then, how it enters the body? See, during sexual intercourse, there might be minute tears in the epithelium. Okay, during the sexual intercourse, there might be minute cuts in the epithelium. From this minute cuts, this Troponema pallidum enters inside. After entering inside, it divides locally in the epithelium itself. Underneath the epithelium, it divides locally and forms a hard substance like formation and forms a hard substance like formation. So what is this hard substance like formation? Why it is formed? See, we know that after there is a cut, there will be pus formation, right? But here there is no pus formation. Why? Pus formation usually occurs. Pus formation usually occurs when there is excessive neutrophil activity. When there is excessive neutrophil activity, there will be pus formation. But here there is excessive lymphocyte activity. There is excessive lymphocyte activity, so there is a hard mass like formation. This is known as chance. This is known as chancre. Okay. Now what happens? Okay, it entered and divided locally and form the chancre. Then next what happens? This now here it is known as primary syphilis. After this, it enters the blood vessels and it also enters the lymphoid organs. It also enters the lymphoid circulation. Okay, after it entering the blood circulation, what it does? It causes fever. It causes rashes, and these rashes are very generalized type. But there is one most effective sign which we can which we can understand that syphilis might be the disease underlying to it. That is rashes on palms and soles of foot. Rashes on the palm and soles of foot, we can see rashes, and which might point towards the syphilis. Okay, and after entering this circulation, it is called secondary syphilis. Here we finished primary syphilis, here we finished secondary syphilis. Okay, in the secondary syphilis, these are the things which happen. Then we come to latent syphilis. In latent syphilis, the syphilis causing bacteria, the Troponema pallidum, goes into dormant stage because the immune system suppresses it. Okay, there are no signs or symptoms in the latent syphilis because there is no activity of Troponema pallidum. The immune system suppresses it. Okay, then in tertiary syphilis, what happens? Years after the syphilis had occurred, years after, it will again show its effects. How? In the CVS, see, think this is the aortic wall. The artery supplying, the blood vessel supplying the aortic wall will get destroyed, and there will be bulging of the artery. This is known as aneurysm. Okay, this is known as aneurysm. Okay, then aneurysm we can see, and aortic valves damage also we can see. Okay, then in the CNS there is prolonged syphilis in the CNS. In the CNS neurons, what happens? See, the damage due to the damage in CVS or CNS is not because of the toxins produced by the Troponema pallidum, but because due to prolonged infections, there will be more and more inflammations. So due to that, the actual problem comes. Okay. Then in CNS syphilis, what happens? There will be neuronal damage, right? So there may be dementia, loss of memory, dementia. Then there may be loss of sensation. There may be loss of sensation also. Okay, these are the CNS related symptoms. And one more thing is gumma formation, GUMMA formation. What is this gumma formation? This is, see, in the human body the Troponema pallidum is spreaded, right? So what it makes is, mainly in the liver, in the pleura of the lungs, what it makes is it attracts the lymphocytes and forms granulomatous tissues, granulomatous substances here in the liver, in the pleura. So this is gumma formation. Okay, these are the 4 stages. First one, primary syphilis, secondary syphilis, latent syphilis, and then tertiary syphilis. And we are going to discuss about the lab diagnosis of syphilis. Okay, lab diagnosis. Okay, in the lab diagnosis, see, first we will go with specimen collection. In the specimen collection, we can take first what we had seen after the syphilis, Troponema pallidum enters. We saw chance ray formation, right? So we can take the sample from this side. Then where it goes into the blood, we can take the blood sample. And we also have, if CNS symptoms are there like dementia, we can take CSF sample. In case of congenital syphilis, what is congenital syphilis? In case of untreated syphilis from mother to child, it might pass. Okay, mother to fetus. We can take cord blood, or also we can take cord blood we can take, and infants blood. Okay, we can take cord blood or infant's blood. This one also we can take. Okay, these are the specimens. Then after specimen, direct detection. Okay, how can we detect directly? See, after we collect the sample, if we check it under dark ground microscope, see, think this is the slide of dark ground microscope. In the dark ground microscope, the Troponema pallidum appears like this. Okay, the background is dark, and this one I will draw with a different color. It is not red color. Under dark background it appears white. Okay, white in color. It is not red. I just drawn for the visibility sake. It's not red. It's white in color. And under dark background it appears white. Okay, what are the features then? The features are It is very thin. It is actively motile. See here, it is thin. So due to its thinness, it can't take normal stains. Normal stains, like we can't do normal gram staining because it is very thin. Okay, so it can't take the stain properly. So it is avoided. It is actively motile, and then it is croc screw shaped. It is thin, actively motile, and cork screw shaped. It appears bright against dark background. Okay, this is the dark ground microscopy and features. Now we'll discuss the second one, that is silver impregnation. We are going to discuss about silver impregnation. See, in direct detection methods, we have two. One is dark ground microscopy and second one is silver impregnation. Now we are going to discuss about the second one, that is silver impregnation. The stain we are using is Fontana. Okay, this is the stain we are using. Here, if this is the slide of it, against light background, it appears black. Against light background, it appears black and shiny. It appears shiny. Okay, silver makes it shiny. The stain uses Fontana. Okay, silver impregnation also over. Then we come to the serological tests. In the serological tests, what are all there is? One is non-troponymal test and troponymal test. In the non-troponymal test, I will make a concept clear. See, if this is the cell wall, whenever this cell wall is broken, due to what Troponema pallidum, this cell wall is made up of lipids, right? So these lipids will leak out. One of those lipid is cardiolipin. What happens to this cardiolipin? The immune system of our body detects this cardiolipin as antigen and produces antibodies against it and destroys it. And these antigens are circulating in the blood. Okay, whenever we take the patient's blood and we add into cardiolipin containing substance, cardiolipin containing container, what happens? There will be floccules formation. See, whenever we are putting the sample of the patient's blood with with antibodies against cardiolipin into cardiolipin containing container, the antibodies will catch the cardiolipin antigen like this, will catch it and form clumps. These clumps are called floccules, and this process is called flocculation. CCLUTION. LATION. Okay, this process is called flocculation. Okay. Now, this whole process in non-troponymal test is known as VDRL test, Venereal Disease Research Laboratory Test. Okay, this is very important 3-mark question. VDRL. Okay, this is non-troponymal test, and we'll go with troponymal test. In the troponymal test, see, the test name is Troponema pallidum hemagglutination assay. First, we'll take RBC in the lab and coat it with Troponema pallidum antigens, and we will add it with patient's serum. Whenever added with patient serum, there will be agglutination of the blood, agglutination of RBC, which represents a positive Troponema pallidum hemagglutination assay, which comes under direct troponymal test. Okay, these are the two tests, non-troponymal and troponymal tests. Okay, by this we had completed the lab diagnosis also. There is no specific artificial culture media for this Troponemia pallidum to grow. These are the important ones. Okay, then I had already told, right, the treatment will be benzathine penicillin G.